basically for rice, I'm going to present you now what some. I hope I will not be too redundant compared to what you have been received already. But I'm going to spend some time to talk a little bit about rice, the rice growing, and then the origin of rice. Some key indication on rice genetic improvement, but I'm going only to focus on classical breeding. I'm not going to talk about genome editing and CRISPR-Cas9 technology. That could be done on the next classes if needed, but this I'm just going to limit today for the classical breeding. But then I'm going to talk about biotechnology in terms of marker-assisted selection. And we are going to view the different type of marker-assisted selection. Although they have different generation of tools, they're still all in use today. And then I'm going to illustrate some rice breeding activities at CIRAD and with the partners, reviewing the targets, the current and historical perspective, and our perspective. The current and historical partnership and our current perspective. And if we have a little bit of time, I will talk to you about rice in France because many people from the tropics wonder if we produce rice in France. And so I have a few slides on the rice that is produced in southern part of France in Camargue. So first, a little bit, I hope I'm not going to be redundant to what you have been already studied, but a little bit of review on rice and the grass family, the Poaceae. And a very important family, the Poaceae, which is here appearing on this tree a very long time ago, about like more than 100 million years ago, and that encompasses the Oryza group here, Oryzadae, and also some other important grass species, which is here the maize and sorghum, and then the wheat and other important crop species of today. What is interesting to see on the, I don't know if you see my mouse, but on the right insert, the grass genome synthetic comparative genetic. So this is a key contribution in the late 90s, showing in the center, the inner circle, the rice genome, and then on the most extreme center circle, the old genome, and the centenary, which is like the similar position of key gene of interest across the different grass genome. And we see that the rice has this particularity of being called a model crop because they have a very small genome with 12 chromosomes that makes a very interesting model for studying other more complex like oat or wheat or maize genome. The grass family, so the genus Oryzae, encompasses 27 species. Among those, two are cultivated species. So Oryza sativa, that you all know from Asian origin, the Oryza glaberrima, which is from African origin, and those cultivated species have a very small genome of 410 megabase and is a diploid with 12 pair of chromosomes. So you have here a tree with all the wild species. Some are tetraploid and have a different genome, the B, the C, the D, the E. So there's a very wide diversity of wild rice, and you can see on the lower picture the phenotype of those wild species that has been published in 2013, and that really represent a very diversity of these species. So the rice origin, Oryza origin more than 15 million years ago, and the Oryza sativa about 500,000 years ago. So you have the Oryza sativa indica and japonica, which are the main cultivated subspecies of rice. Um, if we look at the diversity among the cultivated Oryza sativa, so as I said, you have the japonica, which represent 32%, and the indica 48%, which are the main subspecies of rice cultivated across the world. So the indica has a pan-tropical distribution, and the japonica include the temperate japonica as the one that we have in Europe and southern hemisphere in Latin America in a lower latitude. Um, and then you have, so those two are the main, and then you have the house, which represent 8% of the diversity, which are mainly in the Himalayas and Iran and India. We have the very reduced representation of floating rice, mainly found in Bangladesh, and I think I found some in in Thailand and also in Cambodia. There's in Kardi, when I visited Kardi, there was a floating rice that was more than 6 meter height. And then you have the basmati rice, which are really localized to the Himalayas from Iran to Myanmar, and with different type, the sadri and the basmati. So back in the 90s of 86, you had with Isosam a diversity mapping of the rice, of the cultivated rice. And you have different type of grain, but also a diversity that was expressed with Isosam. And what was interesting is that in 2014, so more than 30 years later, you had exactly the same distinction of the different cultivated rice, but done with the 3000 rice genome project with the resequencing. So basically, we fine-tune the distribution of the diversity of the species Oryza with more advanced technology. So the rice ecosystem, so you have different way of cultivating rice from the upland to the submerged rice. And you have the different subspecies of rice, the tropical japonica and the indica, which are part of the Oryza sativa, that have adaptation to the different gradient of cultivation of rice. And this gradient of cultivation of rice is basically the water management. So in the upland where there's no standing water, the Oryza sativa tropical japonica will be more adapted versus in the submerged area or irrigated area or lowland, it will be more prone for the indica type. And you have in the global representation of the distribution of rain-fed, lowland, and irrigated rice, which are the three main rice growing system. So in Southeast Asia, you have mainly irrigated rice, but you see that in Latin America, you have mainly rain-fed rice. So the production of rice, as we know, is largely skewed and with a big presentation in Asia and Southeast Asia. And but the beauty of rice is that it's cultivated everywhere. It's a tropical plant, but that also accepted a very wide range of temperature from 13, 14 in a temperate zone to a maximum, sorry, of 40 degrees in the very hot areas of southeastern Asia or in West Africa. The latitude of cultivation is very wide, from 4 degrees south in Argentina to 53 degrees north in China. It grows from the delta area to the very high hillside of Nepal with 3,000 meter, and it tolerates very different type of soil, from very acid pH to very basic pH, with different kind of stresses such as salinity, toxicity, aluminum, or iron. So it's a very widely cultivated crops that feed a very wide proportion of the world. Indeed, here you have a map of the consumption of rice. All what is blue is really predominantly rice consumption, and it goes to the purpling when there's a mixed consumption with corn. The green color is mainly wheat-based diet, and the red color is mainly corn-based diet. So if in France we eat in average 9 kilos per year per habitant, in Southeast Asia it goes to 270 kilo per year per habitant. So you have very wide distribution of consumption of rice, but this is that what makes rice the second most important crop in the world. That was a small introduction on rice. Now about the genetic improvement. So I don't see you. So if you have any question, please don't hesitate to interrupt me during the presentation. In terms of genetic improvement, this is a representation of a scheme for plant breeding program. The plant breeding program starts with the species diversity and locally adapted material. So what we want is to exploit the genetic diversity of the species, but to also look at what is locally adapted to see if we can combine those two pools of germplasm to develop new varieties. So it's always a search for complementarity, and then we evaluate those material through phenotype, but also to the pedigree. We know the ancestry of the material. And then we make crosses. We develop new genetic diversity, and this new variability is evaluated and is selected. So we have a process of structuration of the diversity through genetic fixation. So basically, once we develop the diversity, we want to fix the genotype. So there's a step of genetic fixation, but at the same time to select. We select the best candidate to follow new step of evaluation with the phenotyping of the target traits for the environment that we plan for the expected environmental condition of our breeding output. And then we follow the selection of new entries and further evaluation. But at one point, when the number of selected entries is reduced, then we can phenotype in the targeted site in order to really see what is best fitted for the environment. And then this new combination of traits and improved performance is further evaluated to get new varieties. So this scheme of breeding is general for many crops, and this is general whatever the methods we use. We create variability, we fix the variability in order to make sure that the variety will be fixed and breed through, which means that you will reproduce exactly the same genotype from generation to generation, and then we develop new varieties.